Okay, so in this video we will talk about the remaining part of the photosynthesis. Basically, we will cover from 1.2.5 till 1.2.8, but overall I will review also 1.2.1 till 1.2.4. Okay, so first of all we are going to talk about the photosynthesis. So you remember photo means light and synthesis is the preparation of the glucose. So this is the formation of glucose in the presence of light. And here is the diagram that summarizes the whole process. You must know what's the name of this organelle. So if we talk about the name of the organelle, of course it is chloroplast. And what process takes place in this one is chlorophyll. And the chlorophyll has the parts which are basically, ignore this one, this is not okay, don't ignore. Sorry. So basically if you just label it like this, okay, then it becomes granum. And if you talk like one only, it is known as thylakoid. And thylakoid looks green because they contain the pigment which is called as chlorophyll. Now from here onward we will talk about the equation of the photosynthesis. So if we talk about the equation of photosynthesis, we know that H2O plus CO2, you can see these arrows which are going in and they give you the one which are going out. Okay, so we can easily write over here that this gives glucose C6H12O6 plus O2. And we know the rule of balancing, just put 6 with each except glucose. And you should know the names as well. This is water plus carbon dioxide. Carbon dioxide gives you glucose, sugar, or whatever it's oxygen. Now the most important thing that you need to see over here, the reactants of the photosynthesis are H2O and CO2. So these two are the reactants and the products are over here. The process takes place in the thylakoid, that's called as light reaction because it requires the light, and on the Calvin cycle it is dark reaction or light independent reaction, and the organelle is labeled as chloroplast. This is the organelle, but the liquid inside this one, of course, where the Calvin cycle takes place, it is called as stroma. The thylakoid produces two major products, like the light dependent reaction produces two major products which are ATP and NADPH, while on the other hand Calvin cycle produces the reactants for the light reaction. So we go into the first KPI 1.2.5, explain using visual. So of course you should have a diagram or a graph, okay, or any picture or any data that you will use to explain the main events that occur during the light dependent phase. What are the basic things that you need to know is the role of the light. Okay, the role of the light is basically involved in splitting of water. Okay, so splitting of water, because this is the light reaction, so that's process taking place over here. So you have water, it breaks down into oxygen, protons, and along with that you should know plus electrons. So if it is 2 protons, so you will have 2 electrons. If it is 4 proton, you will have 4 electrons. So these are the major products of the first step, that when the light hit. Now you should be knowing that this process is called as the light dependent phase. Okay, light dependent phase. Okay, and in this one, this starts with PS2. Okay, so this is PS2. The first part where the light hits is PS2, and the second part where the light hits is PS1. So from PS2, what happens? The photolysis of water, photolysis lysis of water take place and the electrons get excited. They move from PS2 to PS1 through this chain that's in between over here, and this chain is called as electron transport chain. So this is electron transport chain. Okay, and then the electrons reach over here in the last one, this area, the purple one that you see. This is ATP synthase. This is called as ATP synthase. So when we talk about the ATP synthase, you should know that what do this ATP synthase do. It helps to join the ADP, you can see over here, ADP with PI to give you the final product, that is ATP. So four major things that you must need to know over here. Five actually, okay, six actually. So first of all, it is PS2. Where is the PS2? Then where is PS1? Then where is okay, so let's make them. So there is PS2, then after that we have electron transport chain, then we have PS1, and then we have ATP synthase. Okay, so now what happens on the PS2? Photolysis of water. Okay, and the electron transport chain help in the production of ATP and NADPH, which are the energy storage molecules, and the PS1 further helps in the transport of electrons by absorbing the light. Okay, and they help in the conversion of the final this, and ATP synthase helps in the production of ATP by a process that we will learn later, that's called as chemiosmosis. Okay, now one more thing you need to see over here, that the protons are over here in the thylakoid. So when the water breaks down, the water is breaking down in the thylakoid and the protons get accumulated over here, and ATP synthase transport these protons. You can see this arrow out, it's going out towards the stroma. So on this side it is stroma. So it's listed over here also. So the protons move from the thylakoid to the stroma by the help of the ATP synthase, and in this process ATP is produced. What is the main role of the water in the light phase of light dependent phase of the photosynthesis? So you should know what happens over here. Okay, so to provide carbon atoms for glucose production, of course no, because carbon comes from what we say, from the carbon dioxide. To absorb light energy and transfer it to chlorophyll, that is possible, but they don't transfer it to chlorophyll. To store energy for use in the, no, of course no, they don't store energy. To supply electrons and protons for ATP and NADPH production, yes, that's the correct one. So light dependent phase, as I told you, they help to produce what you can see over here, ATP. Okay, they help to produce ATP, sorry, they help to produce ATP and NADPH after the movement of electrons and protons. So D is the answer. Use the diagram to describe the details of electron transport chain, ETC. Where does this happen? In the thylakoid membrane. So you can write it capital ETC. And I told you already how does the ETC look like. So this is ETC, okay, and the middle one over here that links the photosystem 1. Okay, let me change the color again. So it links the photosystem 2 with photosystem 1. So okay, so you have to understand, and then also know that the water breaks down into three major components. You can see the three components over here. There are protons, there is oxygen, and there are electrons. So three components are produced when the water breaks down, and this helps by the light. So light energy is absorbed by the photosystem 2, and water molecule split into oxygen and it is released from the cell. There are protons that stay in the thylakoid and an activated electron that enters the electron transport chain. So these are the three major products that are produced over here. After that, these move to the photosystem 1. Okay, so this is photosystem, this is PS2 and this is PS1. The electrons move from here, go all the way through the electron transport chain, go to PS2, where they are further transported out into the stroma and help in the conversion of NADP plus H plus into the final product, that is NADPH. This is one of the product. We have another product that is called as ATP, and that is done by this enzyme. This is ATP synthase, okay, and that converts the ADP into ATP, of course with the help of the proton gradient. And this process is called what? Chemiosmosis. So chemiosmosis is a process in which ATP is produced by the help of the proton transport from the thylakoid space into the stroma. So they move from the thylakoid and go into the stroma. DCMU is an herbicide that blocks the action of the electron transport chain. So electron transport chain is not working. Okay, when the electron transport chain is not working, you should know who will be affected. ATP will be affected and NADPH, because these are the two direct products of the electron transport chain. So which of the following is direct consequence of using this one? So you should know, as we told you, so these two will be affected. Decreasing the production of ATP and NADPH, yes. Inhibiting the splitting of water, no, because this takes place in the PS2 and the electron transport chain comes after PS2. Blocking the light absorption, no, that's not possible. And breaking down the chlorophyll molecules, no. So of course A is the only correct answer. Next KPI. Describe the importance of the chemiosmosis. I told you already, chemiosmosis involves the movement of protons. Okay, before we go into this detail, chemiosmosis means ATP synthesis. How this ATP synthesis happens? By the movement of protons across the thylakoid membrane through channel called ATP synthase, creating a proton gradient that drives ATP synthesis. So where do they go? All these they go. These protons move from thylakoid, because they are in the high concentration over there, into the stroma to balance the concentration. Okay, these protons move from thylakoid to stroma. So chemiosmosis is a process in which ATP is synthesized by the movement of protons from thylakoid to stroma by a channel called ATP synthase. This movement drives the ATP production. What is the main importance of chemiosmosis in generating ATP during photosynthesis? So it directly breakdowns? No, of course there is no breakdown. Okay, so focus on the keywords. It uses the proton gradient. Yes, we discussed over here. It uses the proton gradient across the thylakoid membrane to power ATP synthesis. So chemiosmosis, its meaning is ATP synthesis by the help of the proton gradient. So yes, this is the answer, but we will see the next also. It pumps protons. It's not the importance of chemiosmosis. Who pumps protons? It is ATP synthase. Okay, it pumps protons into the stroma. That's wrong, because ATP synthase pumps protons, creating a proton gradient needed for carbon fixation. No, needed for ATP synthesis. It transports the electrons. No, so only B is the answer. The last KPI 1.2.8, explain the main events that occur during the light independent phase. So where does this phase takes place? Stroma. We know that what is the product? Glucose. We also know that what goes in is CO2. How many CO2 goes in? 6 for a complete photosynthesis reaction. and the formula for the glucose is C6H12O6. Okay, but every cycle you see one molecule goes in. The cycle repeats 6 times to give you this one. Now, in this cycle, we divide it into 3 halves like this, 1, 2, and 3. So the first half over here, this is the process in which CO2 is fixed into a 3 carbon compound. Okay, and this process is called carbon fixation. Okay, we label it as step number 1. So when we go to step number 2, it is called as reduction. It is reduction, and in this process these carbon molecules rearrange to make this carbon molecule to be recycled, and at the same time you produce G3P, like a 3 carbon compound. So not all of these are recycled back into our UBP, but some leave the cycle as G3P. And the last one, step number 3 over here, is called as regeneration. And who is regenerated? RUBP. RUBP. And there is an enzyme involved over here in this stage. This enzyme is called as Rubisco. Rubisco. This is Rubisco. So Rubisco is an enzyme which is involved in the carbon fixation, and this is present in the cycle itself. So we have the steps written outside: carbon fixation. Okay, carbon fixation, this is number 1. Number 2 is reduction, and number 3 is regeneration. So you should know who is reduced and who is regenerated. And one more thing over here, you can see C6 ATP are going in, and who is coming out? ADP. And same over here, NADPH goes in, and who are released is NADP plus. Okay, so what are the 3 steps of this cycle? Number 1, carbon fixation. And the enzyme involved in the carbon fixation is Rubisco. Number 2 is reduction, and the reduction mean H will be added. So this H which are coming from here, so you see this is missing over here, this is added, and the process leads to for reduction of the molecules. And the last one is not oxidation, sorry. The last one is regeneration. And who is regenerated over here? RUBP. Okay, and remember, RUBP is a 5-carbon compound, and 1 carbon comes from here, from this, so they make 6-carbon compound, but they rearrange themselves to further molecules. Now moving to the next one, what is the primary role of the Calvin cycle? I just told you what is the role of the Calvin cycle without looking over there. So CO2 is being fixed. Okay, and what do you produce? What is the main ultimate aim? Is the glucose. So absorbs light energy? No, of course it's a light independent reaction. Fixes CO2 to produce glucose molecule? Yes. Makes NADPH? No, it uses NADPH. And produces oxygen? No, because it produces glucose. Which of the following is the number of carbon dioxide molecule? Okay, we know that carbon dioxide molecule that are 6, right? That you need minimum to produce glucose. Okay, so 6 carbon dioxide are used to produce 1 glucose molecule. A biologist exposed 2 groups of the plant's experimental group for 24 hours. Group 1 received the adequate light and CO2, and group 2 received light but no CO2. After 24 hours, both the groups were tested, and group 1 showed high glucose production because group 1 was in light and it was having CO2, and it was like given all other supplies. The group 2 showed almost none. Which of the following explains why group 2 failed to produce. So we know that there is no CO2. Okay, so simply there is no CO2. So focus on here. So chlorophyll becomes inactive without carbon dioxide. No. The Calvin cycle depends on the carbon dioxide. Yes. To form organic molecule. Yes, that is the glucose. Without CO2, carbon fixation does not occur. True. The Calvin cycle requires light. No. Without oxygen, the Calvin cycle, we don't need oxygen for the Calvin cycle. So all other are related mostly to the light independent direction. Light dependent. Use the diagram below which shows the independent phases of the photosynthesis. That's also called as Calvin cycle to answer 7 and 8. What's 7? Identify the step that represents carbon fixation. So of course this is carbon fixation. So it is step number 1. Explain what is happening in step 2. We know that in step 2 what's happening. The it's called as reduction, right? Reduction. So step 2 is reduction. So when we talk about the reduction, you can simply say that the chemical energy that was stored in the ATP and NADPH is used. Okay, I will repeat again. What's happening in this stage? The chemical energy that is stored in the form of ATP and NADPH, it is used and transferred to make G3P. It is used to make G3P, and ATP supplies the phosphate groups. What does ATP do? They supply phosphate groups. And what does NADPH do? They supply protons. Okay, so they supply protons and electrons to let the cycle continues, while the ATP provides the phosphate for G3P molecules. Complete the table below writing the stages of photosynthesis if it is light dependent or light independent. Use light energy. Of course, it is light dependent. Produces glucose, Calvin cycle. So light independent. Takes place in the thylakoid, of course, then light dependent. Takes place in the stroma, of course, light independent. Produces ATP and NADPH, definitely light dependent. And uses CO2 is light independent. Okay, now moving to the next question. The student did an experiment and repeat the experiment more times using filters in front of the lamp, and thus through the either red light, blue light, or green light. Only let it. Okay, so color of the filter when the filter was used or no filter, number of bubbles per which bubbles will be produced, by the way. That's a good question. So which process is this one? It is photosynthesis, right? And you know that the photosynthesis releases which gas? It is oxygen. So these bubbles are for the oxygen. This part is okay, but you can you find it by 47 plus 84 plus 80 divided by 3, but don't worry, it's not really important. When you use the filter, it means that that color will be reflected. Okay, so red, number of the bubbles produced over here is 48, 48, 42. So these are the numbers of the bubbles. What we need to know over here, the most important thing is like what's happening with these lights. Okay, so you should know when you are using these colors of the lights, what's happening. So when there is no filter, all the colors are falling on the plant and they absorb specific wavelengths. So the process of photosynthesis takes place very fast. How do you know the process is taking place very fast? It is because of what? It is because of the photosynthesis. And what does the photosynthesis will produce? Bubbles of oxygen. So why it is important and why it is not valid to use the number of the bubbles. Okay, so what are the reasons? So let's do first why the scientists measure oxygen bubbles to check if the process is taking place or no. So let's go for the importance. What's the importance? Basically, the importance is that the number of bubbles, the number of bubbles uh, number of bubbles is directly related to the rate of reaction. And which reaction is this? Photosynthesis. Okay, so this is how it tells us. So there is a direct relationship. Okay, remember that between the, like, why it is important to in an experiment to measure oxygen bubbles, because the number of the bubbles produced is directly relation to the speed of the process. The higher the speed, the more the number of the bubbles. Now, why it is invalid? Why it is invalid to measure the number of the bubbles? What can be the possibilities? The possibilities are miscount. So if someone miscount the bubbles, they will make the results wrong. Maybe the bubbles are of different sizes, so the bubble size can be different. So there can be miscount. Okay, maybe miscounted, maybe the number of sorry, the bubble size will be different. And one more thing, might be these bubbles are not oxygen. Bubbles are not oxygen. So this is maybe another process taking place over here, like respiration and other process which helps in other processes. They release different gas, but since it is coming as a bubble, so you don't know what's happening. Okay, and that's it for this process. Wish you all the best, that you do your exam in a nice way. Thank you.